Thank you very much. It's a really great pleasure again to be here after about a month and a half away in the UK. So I'd like to talk about time-lapse. It's not a new technique anymore, but about the hype, the hope that it has in the experience, especially from this hospital, of the way that we use it in our everyday workload. And I'd like to start off with this photograph because we have a craze for taking photographs. I still can't get over this selfie stick that people are using at the time. But we love technology. We love anything new, and especially photographs. And with time-lapse, it's taking a series of photographs, putting them together, and having a video of embryo development. Very popular photographs. In 1 month, there'll be 9 billion photographs which will be uploaded to Facebook alone. So we like innovation, especially embryologists. This is what drives us. This is what makes us more successful and, of course, more commercial. But from Facebook to another face, of course, this is the deity Ea, the goddess of health, and it's our health group logo. And it's a statue, and it's a sculpture from about 350 BC. But it's only in our recent history that we've discovered that the markings on her face were put there deliberately. Before, people thought this was due to time or due to the sculpture process. But these were put there because, in fact, even the goddess was susceptible to pox. And surviving this disease means that she was very healthy. So perhaps taking a closer look at something can give us an insight into the health. And that's exactly what we believe time-lapse can do. But the premise is the same. Whatever you believe, time-lapse was brought into the lab, 2 things: less disturbance for the embryo and more observations. And at the end of the talk, that will, of course, stay the same as well. Before I start, any new technology goes through what Gartner, an IT company, call a hype curve. And it's divided into 5 sections. There's an innovation trigger. There's a peak of inflated expectations. When these expectations aren't reached, there's a trough of disillusionment. And then slowly, as a new technique is accepted, it finds there's a slope of enlightenment when we're using it in a natural way until we come to a natural equilibrium where we know that use for that technique. And we can use this also in science. We can perhaps use this for PGD and PGS, but we can certainly use it for the time-lapse initiative over the last few years, where there's been a lot of papers involved with this technique. So if we start off with the innovation trigger, what was there? Well, there was the disillusionment, if you like, from the metabolomic study. Everyone thought that we could select embryos by looking at the metabolic profile. And of course, in the aftermath of the disappointing papers which showed that PGS using fish did not help embryo selection. So in this void, if you like, the first time-lapse paper was by Payne in '97, and he made a makeshift time-lapse machine by just encompassing an inverted microscope, and he looked at polar body rebuttal. He looked at when the polar bodies came out of the oocyte, and that led to a new interest in this technique. Pribensky, a friend of ours at Matera, who's been here several times, he had the first paper on using time-lapse to singly select the best blastocysts for transfer, and the first pregnancy using time-lapse for that. After that, there was a plethora of papers for time-lapse, even more reviews than actual hard data at that time. And then with the use from EV, a very large group in Spain, there were lots of papers that came out that generated a lot of data, and there was suggestions that we could find a calculation, an algorithm, which could perhaps select the best embryos. And then at the height of the hype, perhaps misleading was the use of, perhaps it can be used for aneuploidy screening. And then, of course, the backlash happened. There was no randomized controlled trials at the time. Even when they came, there was a negative feeling, especially for units which didn't have the time-lapse technology, that perhaps it was just an expensive toy. And I think where we are at the moment is to have or to not have time-lapse. I'm a fan of time-lapse. We've used it every day here in the lab at Matera. And I think that it will gradually become to every unit. And there are many reasons which I will now describe. Because at the moment, what do? We look at static observations. We can look at the maturity, which is related to the stimulation protocol. We can look at the fertilization. And many people have looked at the axis for the pronuclei. They've looked at the pronuclei morphology to try and choose the best embryos. We can look statically, again, in a single view or twice-a-day viewing of fragmentation. This is a very good indication of embryo stress. The most viable way of looking at embryos and how viable they are perhaps is the cell number. Of course, that was from the start of the IVF, and that's our most accurate marker. In a static observation, we can look at blastocysts, so often the endpoint of many studies, and a sign of good lab conditions. And perhaps indirectly for aneuploidy, we can look at the amount of multinucleation, which can affect over 40% of in vitro embryos. But all this can be done in a much more objective way with time-lapse. We can look at it in a dynamic manner. We can go back and assess the details. We don't have to be there. We don't have to take them out of the incubator. There's a continuous observation of the rate and the manner of cell division. We can deselect embryos which we believe are dividing outside the range of what we say are our milestones. It's also a very objective way of scoring embryos. Sunval showed that even if you're a novice embryologist or an experienced embryologist, you will agree on the number of cells, and you will agree more likely than not that the embryo you've scored is the same from an experienced embryologist or not. So that's it's taking the subjectivity out of embryology as well. And it's a great communication aid. It's a great communication aid for the doctors, for the clinicians. It's also very important to demystify all this technique with the patients as well. So this is just a quick video to show what we're talking about. We culture in a wow dish, and we're taking photographs so we can see the embryo dividing at certain stages, and we're marking those stages on a graph. I'll just let it run for a few more moments now. And then, of course, at our own leisure, we can compare the embryos. Many of these factors we would miss. We can either review them the next day or even see them online if we wish to be that way inclined. So that's the whole of the embryo history, if you like, on video. And we want to see a stepwise motion here. In other words, from one cell to 2 blastomeres, to the start of the 3 cells, and then to the 4 cells. And we know that the embryo needs time to organize itself. We know from the start of IVF with Edwards that early cleavage is better, but we now know from time-lapse that too early cleavage is detrimental. Even without time-lapse, a paper last year by Hill showed that fast development on day 3 is a poor indicator for viability. So we want to see a long phase for the second cell cycle when the embryo is at 2 cells, lots going on in that time, a quick conversion rate from 3 cells to 4 cells, that shows that the blastomeres are in sync, a third cell cycle where it seems to be static at 4 cells, and then a quick division rate for 5, 6, 7 cells, and that will go on until it becomes a blastocyst. The cell needs time to do this work, and we know that if it's done quickly, then we'll get incomplete nuclear replication, and the duplication of the cellular organelles will not be distributed correctly amongst the embryos. And that just shows again what we look for. We look for changes in the cell cycle and the transitions. What does this mean in practice? This means the Goldilocks effect, where we have the embryos here, we have 9 embryos here, we have their time profiles on the right, and we can see some which are too quick, some which are too slow, and some which we believe are just right. Now, is there such a thing as perfect timing? And I believe external factors weigh very heavily on this account. I think there's never going to be a perfect timing event across the board, and that's why the algorithms, I think, haven't been able to be replicated in papers from Conahan and papers which we first got from Messenger. But this modified graph showing all the work which is being done by the leaders here from 2013, you can either use this graph to dismiss time-lapse or to, in fact, encourage its use. What I would say is people are looking at different time events. They're looking at the first day until it gets to 5 cells. People are looking at perhaps these early cellular divisions. But what's encouraging is we're homing in on very important time events. And these would be perhaps the 2 to the 3 cell stage, the 3 to the 4 cell stage, and especially the time to the 5 cell stage as well. So all this work is going on. There's a lot of data coming out. And as these companies have been merging recently, I think we're going to come out with some really interesting data soon in the future. There's a British saying, which is integrity is doing the right thing even if no one is watching. Well, that, of course, is true for embryos, and we don't see everything. And there are things that we miss when we go home at night. And obviously, we can't take the embryos too often out of the incubator. So when we first started to use time-lapse here at Matera, we took all the data that was available for the papers, and we looked at the intervals, and we looked at the milestones. The intervals would be, for example, the time when the pronucleide disappears to the 2-cell stage, or the 2-cell to the 3-cell stage. How long does that take? But also the milestones. At what time do they become 2 cells? At what time do they become 4 cells? And so on. And we were very encouraged by its uptake in the lab. It invigorated an old embryologist like myself, but a very keen uptake by the young embryologists. We spend about half an hour, perhaps, on each patient before the embryo transfer, fine-tuning and checking which embryos are best for transfer. But time-lapse speaks our language. It speaks morphology, the language of embryologists. It's not like looking at bands on a polyacrylamide gel, if it's to do with PGD, or looking at a profile for CGH, nor is it looking at a number which a spectrometer spits out in the case of metabolomics. We all know about morphology. We know what we're looking for. We just have to time these events and exclude embryos which we believe are behaving abnormally. So we embarked on a study, and this was with collaboration with the Atacon Hospital, where we looked at a prospective cohort study it was a 4-month study. 249 embryos were divided either with time-lapse or with a normal conventional viewing. And we found there was a higher clinical, ongoing, and birth rate using the time-lapse system. And that was true across the board for each age group that we looked at. We also found there was important time ranges which were significant. And when breaking down into age, we saw it was significant, the 2 to 3 cell stage and the 3 to 4 cell stage, the time when they start this second round of division, if you like, that was significantly important to their pregnancy success. Another way of looking at it is we looked at the in-range embryos that produced pregnancies, and we looked at the embryos which were still in range but had a negative outcome. And if we look at the blue line here, which is the patients who became pregnant, there was a 10 to 20% better chance of leading to a pregnancy if all these embryos were in range at that time. This, unfortunately, was only seen in the early cell divisions, so at the 2-cell stage, the 2 to 3 cell stage, and so on. Towards the later cell divisions, the selective advantage was not seen. And looking at the papers and looking at the trends, what was happening was that we could not convert other people's data from other labs to what we had in our lab. And a paper by Kamaru said that each lab has to define its own data by its own population set. So we tweaked our data, as most people do now, and we changed the time between 5 to 6 from less than 6 hours, we narrowed that to less than 3 hours. And again, we then showed there was a selective advantage to choosing the embryos which fell in that time frame. And I think many of the people have seen this where they haven't been able to convert some of the time intervals from lab to lab. Secondly, I'd like to talk about aneuploidy. There's a lot been talked about aneuploidy, and can we select using these morphokinetic viewings of the embryo? Well, very early on, we started to look at our embryos, and this is a screenshot of just one timeline, and these dots are time events. And we were looking at embryos which took a long time to get from the 3-cell stage to the 4-cell stage. And very luckily, we were able to collaborate with Lena Florentine from Alpha Lab, and we sent these samples to be checked, and invariably these were chromosomally abnormal. And the majority of them, of course, were chaotic, which we can see here by not being a 1-to-1 ratio, but to see various anomalies from the mean along the side. We took it no further. This was elaborated on, though, by a colleague, Steve Davis here, who had a paper at ESHRE, which was awarded, which was specially awarded at ESHRE, and he saw that the delayed cleavage events, especially between the 2-cell and the 4-cell stage, can be identified as aneuploidy. And the amount of aneuploidy, or the more aneuploidy that the embryo was, the longer these time events happened. So, for example, a euploid embryo would divide quicker from the 2 to 4-cell stage than a single aneuploidy, which would divide quicker than one which had multiple aneuploidies. Other work using aneuploidy, Natalia Basile from the EV group, they modified their data for using the algorithms where they had a coding system for each embryo, and they could show that they could classify groups of embryos to select the most euploid embryos. And finally, a very controversial paper by Nicole Campbell, who started perhaps the backlash of being used in aneuploidy screening, was an aneuploidy risk model. It was probably a flawed paper. It didn't take into account age, and it only looked at blastocyst transfers there. She had a risk model which would say what are the chances of the embryos being aneuploidy. So I really don't believe that we can look at the subtle differences with aneuploidy, with time lapse. Perhaps it can help us to select a few of the embryos out. But just to give you an example, we are talking about everyday use, and this is an example we have from the lab here at Matera. This was the patient who had PGS, but she also had time lapse. So it's a good example just to run through what we would see in this event. She had 8 embryos. I should say she was 36 years old. She had 2 miscarriages. She had a normal carrier type, as was seen. So she had 8 embryos. And if we just took it on time lapse alone, we'd have a look at the timeline profile. But we'd probably, by time lapse, have chosen among this cluster which we believe is the formula for selecting the best embryos. And we would have neglected the ones which were outside this range. We biopsied, we did the biopsies at a cleavage stage at this point in time, and we waited for the results. While we waited for the results, it was our practice to go to blastocyst stage. So from these embryos which we biopsied, 7 out of the 8 went to blastocyst. And when we had the results back, we saw that one of the selected ones was in fact correct, it was euploid, but one which we would have selected out, that was, of course, euploid as well. And that's just to show that I don't think that we can obviously, it's not subtle enough to have a look at these fine euploidy events. Finally, looking at the last embryo here, which didn't go to blastocyst, we found that to be chaotic. And that made us go back to the video and go back to the data and have a look. And this is how we have refined and are always refining our time-lapse data. We saw that the time value for the pronuclei to disappear was very, very short indeed. It was less than 20 hours. And doing a bit of searching, we saw that Azzarello, a few years earlier, had a paper showing that it's a very negative viability value if these pronuclei disappear before 20 hours. So that's now incorporated into our time-lapse morphokinetic data. I think time-lapse is here to stay, and I think that until recently, everyone has perhaps some kind of sheet of paper, and they write down what time they look at the embryos, how many cells it was, and so forth. But in a busy IVF unit, we're not going to be looking at embryos at the same time, and we're certainly not going to be doing the ICSIs at the certain time. We'd perhaps like to look at the pronuclei 16 hours after fertilization. Perhaps we'd like to look at early cleavage at 26 hours, and perhaps look at 44 hours and see if there are 4 cells. But perhaps in a busy IVF unit, as I said, we're not looking exactly at the same time. But with the time lapse involved in this, then we can obviously have a timeline, and we can carefully modify our work to look at these exact timing events. So I believe in the future, and this is a prototype for a new system which combines both visual morphology screening with pictures, with videos, and I think we're going to see this in front of a doctor's PC in the future when they're choosing embryos, when they're talking about embryos as well, which are going to be transferred to the patient. So we've gone full circle. Nothing's changed. It's still less disturbance, but more observations. As I said before, there's a greater communication between lab, clinician, and patients. We found a very good pickup from patients who want a DVD. They like to take a DVD away of the embryos which they transferred. It's certainly a more sophisticated way of culturing in vitro embryos. It's been over 40 years, and we're still walking around the lab with a dish of embryos in our hand. It's encouraged the use of one-step medium, and time-lapse special medium is being produced now. It's been driven by this need to less disturbance of the embryo, and that we'll see some good data coming for that in the near future. There's a standardization across the labs. We can compare very easily between embryologists and even unit to unit. It's a permanent record. Even if the couple do not get pregnant, we can go back to this data and we can assess it at a later date. I believe it helps to choose the better embryo for transfer, and certainly which embryos to freeze, by deselecting embryos from a cohort which we're not very happy about how they're dividing, or in fact completely abnormal development. And in recent papers which we've seen, a combination of standard observations and time-lapse has shown to increase pregnancy rate. A fantastic research tool. We can use it now to validate previous studies with objective data. So all the studies before, which were roughly timed, perhaps comparing different types of medium, we can now use this to accurately look at this data again. In a lab which does 100% of time-lapse, we can use this for quality control. It's a very sensitive indicator of lab drift as well. So randomized control trials are now coming, so time-lapse is in everyone's hands, I think, to use it. So my last slide, going back to Gartner's hype cycle, as we reach this slope of enlightenment and we find its place in the IVF labs, there could be some sparks of innovation still. Like I said, we can use it for our KPIs. We can have a look at exact timing events, and we can very accurately look at if there's adverse effects going on in the lab. Lee and Yi are using it. They're looking at the cytoplasmic formation and how it twirls and how it changes just after egg collection. And they have proposed, they know which oocytes are the best to use for ICSI. They've showed that some are going to be aneuploidy, by the way that the cytoplasm is moving. Metabolomics rears its head again. Gardner has showed that there's metabolic events, and glycolysis are closely linked to time-lapse and and morphokinetic cell divisions as well. So there's this resurgence again of the interest of metabolic studies, and we can time this as well. And finally, the holy grail of IVF, which is the lab on a chip. Gary Smith was here a few years ago talking about perhaps having one platform of culturing embryos where we can test it, we can view it, we can do everything. This new paradigm change of looking at embryos, so perhaps that is a step closer to looking at microfluidics on that. So thank you very much.